Pleasure to be here. So indeed, I'm going to talk to you today about how we can detect chromatin modification in cancer using ChIP-seq data and what are the main challenges and solutions. So you probably know why we use ChIP-seq. So we use it to detect binding sites of DNA-associated proteins such as transcription factors, RNA polymerases, and histone modifications or histone variances. This is the main steps. I will tell you briefly what they are for ChIP-seq technique. So what we do, we cross-link DNA of the proteins so that proteins cannot detach from DNA. Then we shear DNA into smaller pieces using, for example, ultrasound. Then we take an antibody which is specific for our protein of interest, for example, some histone mark like H3K27 trimethylation, and we extract DNA protein complexes which contain our protein of interest. Then we purify DNA, we reverse cross-links, we do site selection. And at the end of this step, we have a DNA library, and each fragment in our DNA library was at some point bound by our protein of interest or by our histone. Then we sequence a small piece of these long DNA fragments, usually up to 100 base pairs, and these pieces are mapped to the reference genome, so you see them like this. And if you have a cluster of these mapped reads, then probably there was a binding event somewhere here. In parallel, we do a control experiment. The most often used control is input. This is when we just cut DNA into pieces and we sequence them. You can consider this input experiment as like low density whole genome sequencing. So we do this ChIP-seq to detect histone modifications, and I hope you know what it is. But you know what it is, I'm sure. So you know that histones are proteins around which DNA is bound, and they have tails, and these tails can be covalently modified. And many of these tails, many of these modifications have direct effects on gene transcription level. So for example, the trimethylation of lysine 27 on histone 3, when this mark is found on the promoter region, it means that the gene is probably silent. And if you see this mark H3K36 trimethylation on the gene body, this means that probably the gene is transcribed. So there was a huge effort by the research community to map these histone marks along the genome for many cell lines. And among these cell lines, there are also cancer cell lines. So here you see a list of cell lines in the Encode data set. Tier 1, this means that lots of experiments were done for these 3 cell lines. Many ChIP-seqs for transcription factors and histones, but also RNA-seq, DNase, hypersensitivity sites, many different experiments. And also we have tier 2, for which many of these experiments were done. And among them, you see there are 6 cancer cell lines. If you download ChIP-seq peaks for these cell lines, you will see that they were called using the same methods as were used for normal cell lines. Peak, for me, it's a binding site, or it's the place where we have enrichment in this histone mark. Why you should not probably apply the same methods when you deal with ChIP-seq data from cancer cells compared to normal cells? It's because in cancer cells we have lots of genomic rearrangements. And here, for example, you see a 24-color karyotype for a cell line for lung adenocarcinoma. And each chromosome has its own color. And you see that some chromosomes are present in 2 copies, like chromosome 12, some in 3 copies, like chromosome 7, and some in 4 copies, like chromosome 5 here. You also see translocations. So it may be that since you have different number of regions in the genome, then when you do statistical tests to predict enrichment in histone marks, these tests can be biased. And this is what we can see if we detect these enrichment regions using the most commonly used software like Max and Sicer. So here I show you peaks which were detected by these 2 tools along chromosome 8 And this chromosome has a loss, an 8p region, and again an 8q. And you see that both software under-detected peaks in the region of loss and over-detected probably peaks in the region of gain. And if you look at what methods were used by the Encode Consortium to detect peaks, again, you see this software Max, and also you see another software Scripture. So if you go to the UCC genome browser and you want to visualize peaks, you will see these broad peaks. They are much larger than Max peaks, but Max peaks can be also downloaded using some links. You can Google it. So, and they were calculated by the Broad Institute. So we wanted to check whether there is this copy number bias in these two data sets of peaks calculated by Max and Scripture. And we saw that indeed there is a huge copy number bias. So what you can see here, you see different marks. And you see the coverage by these marks in the regions of loss in orange, copy neutral in purple, and regions of gain in green. And you see that for all marks, repressive marks like H3K27 methylation and active mark like H3K36 methylation, you see much more peaks falling in the region of gain compared to the region of loss. And you see that in some cases this ratio is fourfold. So also if we look how it looks in the UCC genome browser, for example, we go to HeLa cells to the region which is present in 6 copies. This is the read density profile. This is the same region. Here density profile is shown for active mark H3K37 methylation, and here it's for repressive mark H3K27 methylation. You see that peaks which were predicted by Broad Institute or these large peaks, they don't really correspond to the signal you see. So I would put them somewhat differently. And also what you can see from this slide is that there are some peaks that overlap, and this is actually a peak for an active mark which overlaps a peak for a repressive mark. This means that according to them, according to Encode, this region is both active and repressed at the same time. And if you look how many regions like this you have in these HeLa cells, you see that in the region of gain, like 20% of regions which have a repressive mark, they have also an active mark on them. And I don't believe that this is biologically true. So we wanted to reanalyze this data, and to do so, we used a method we developed to analyze the histone modifications in cancer using ChIP-seq. And so the method is called HMCAN. It was developed by Haitham Arshar, thank you, and was published in Bioinformatics last year. In the paper, we compared HMCAN with 3 other peak calling tools, CCET, CISA, and MAX. We did it on simulated data and also on a real data set for H3K27 methylation. I will walk you quickly through the main results of the paper. So first, the outline. I'm not going into details. I just wanted to say that we explicitly normalize for GC content and copy number. And to get copy number, we apply a module extracted from Freak algorithm. This is an algorithm to detect copy number in whole genome sequencing data. But since our input is whole genome sequencing data, we can apply it and get copy number profile. So here you see copy number profile that we can get for HeLa cells from the Encode ChIP-seq data. So after all normalization, the profiles look like this. So again, this is chromosome 8. You have already seen the region of loss and region of gain. And you see that corrected read density profiles are uniform along this chromosome, both for the input and for the chip. And if we zoom in, we can see regions of enrichment. What we do next, we subtract the control density from the chip density using with the constant. And this constant depends on the noise in the chip that we evaluate. And after this subtraction, the profile looks like this. So already visually, you can separate regions of enrichment from a background. To do it automatically, we apply hidden Markov models with 2 states. Yes, we have signal. No, we don't have any signal. So, and it works pretty well. Again, there is no copy number bias, at least visually, in HMCAN predictions. And so we further compared the 3 tools, CCAT, Max, and Sizer, on simulated data sets. And of course, when you do a comparison of your tool with other tools on the simulated data set, your tool is always better. This is what we can see here. So we have like recall almost 1 and precision almost 1, but it's simulated data. So we wanted to compare our tool with others on real data set. Since there was only one tool among these 3 which didn't show copy number bias, we selected only this one for comparison in real data set. But in supplementary figures of our paper, you can find comparison with others as well. So I will show you how we compare HMCAN and CCAT. So first, I should say that, unfortunately for us, predictions were very close. So you see the Venn diagram. So CCAT predicted almost the same as we did. But if we look at the association between these predictions for this repressive mark and gene expression, we are slightly better. So this diagram shows you gene expression for 4 types of genes. First, purple genes and gray. So purple genes, these are genes which have this repressive mark on the TSS predicted by both tools. And this is when there is no mark predicted on the TSS, and we see that gene expression is much higher when there is no mark predicted. This is what we expect. But if we compare genes which have this repressive mark predicted only by one tool, only by HMCAN and only by CCAT, you see that actually HMCAN genes have low expression. This means that we predict more true events. But we missed also some true events, because you see that CCAT only genes, they have low expression than genes which don't have any marks predicted by any tool. Also, we did some validation using qPCR. For example, for the promoter region of this gene, DLEC1, CCAT missed a real signal. And this gene is commonly deleted in various carcinoma, and it was important for this cell line to be able to identify the true signal on the promoter region. So again, in this case, HMCAN showed better performance. This is an image what I had when I analyzed, when actually Haitam sent me data when he reanalyzed Encode data. This is for HeLa cell line. So again, you see 7 different histone marks corresponding the read density profiles, and then in purple you see peaks which are predicted by HMCAN. And this is this region with 6 copies where all other tools had problems to identify correct signal. Then we checked whether HMCAN still have a copy number bias, and we see that for some marks we don't see much of copy number bias, but for other marks it's still there. It may be just because it's easier to be more sensitive in detection of enrichment when you have more signal. It's like when you compare 3 and 6, you are less sure that 6 is more than 3 if you compare 30 and 60. So we still have some I would say we still have some under prediction in the regions of losses. But if you compare this difference in enrichment in the region of gain and losses for HMCAN with what we have for these peaks, which you can download from the Encode, you see that we perform much better than them. We looked whether in the peaks we obtained with HMCAN, we have overlap between active mark and repressive marks, and we see that there is the overlap is less than 1%, so it's good, a good sign. And also, the last thing we did, we compared the coverage we obtain by HMCAN for each mark with the gene expression. So, for example, here you see coverage of transcription start sites by this repressive mark H3K273 methylation for genes which have high expression in red, and up to genes which are silent in purple. On the left you see regions of loss, in the middle it's copy neutral regions, and here you see regions of gain. And you see that for the peaks predicted by HMCAN, the signal is very stable. So, for example, for silent genes, you have almost as many marks predicted in the region of loss than in the region of gain. But if you look at the narrow peaks predicted by MAX, you see that there is a clearly under-prediction of the signal in the region of loss. And you see huge over-prediction of the signal in the region of gain in these broad peaks that you see in the UCSC when you browse the human genome browser. So, for example, you see that for highly expressed genes in red, this repressed mark is found on almost 20% of TSSs of highly expressed genes. It shouldn't be true. This is for another mark, this active mark, H3K363 methylation. And again, you see it's very stable for HMCAN, so no copy number bias. For narrow peaks predicted by MAX, you see clearly that there is some copy number bias, so more peaks predicted in the region of gain compared to the region of loss. And when you look to these broad peaks, you see something strange. First, you see this over-prediction regional gain, but also you don't see this valley that you expect to see on the TSS. This means that they have really a problem with the correct identification of boundaries of the signal. So for some marks, you should not download this data from Encode, and you probably should use our website to download this data. So Hightam recalculated the signal for 6 cancer cell lines from the first and second tiers of Encode dataset, and they should be all histone marks there, I hope, yeah? Yes. If you want to use HMCAN yourself, you can download the package from the website, or you can use it online using our ChIP-seq analysis framework, which is called Nebula, located in France. So here, in addition to HMCAN, we have also many tools to do almost all steps of ChIP-seq data analysis, starting from read alignment, peak calling for transcription factors and histone marks, peak annotation, motif finding. We can construct enrichment graphs around gene TSS, and many other things. So this is the summary of what I just presented. So I hope I convince you that the main challenge in detection of chromatin marks in cancer is the fact that cancer genomes are rearranged, so we have copy number bias. But fortunately, there is a solution, so we can use our tool HMCAN to calculate correct signal. I showed you that Encode peaks contain huge copy number bias and should be recalculated, and you can download the recalculated data from our website for the software HMCAN. I would like to thank people who participated in this work. Hightam, who did the main part for development, testing, and also it was him who recalculated data from the Encode dataset. I would like to thank Amira Kramdi, a master student looking for PhD, who did integration of HMCAN into the Nebula framework. I would like to thank biologists who provided us this data, the data for H3K273 methylation to do comparisons, and Vladimir Bajic and Emmanuel Barrio for financial support. On this, I would like to thank you for your attention, and I will be happy to have your questions.
We have time for questions.
Hi. Really nice talk. We have to use your tool to do all the things we do on cancer cell line, for sure. The question is, have you tested what is the effect of your tool in, let's say, normal cell line? They are never normal. They are immortalized and things like that. So the behavior, when you have not such a dramatic situation of copy number variation, using your tool and another tool like Sizer is going to be similar. Have you tested?
If you want to select a tool to use on normal cells, Sizer is a very good choice. So we compared CCAT, Sizer, and Champkin on normal cells. They perform very similarly. So
Okay.
if you use Sizer, you can continue using Sizer. It's very good. So it's just for cancer cell lines that you can have problems.
Okay, thanks a lot.
Hi, very nice. So if I understood correctly, you're using the ChIP-seq data to find the copy number itself?
We don't annotate exact copy numbers, so we don't say this is 2 copy region,
Right.
this is 3, this is 4. We do normalization, segmentation, and for each segment we calculate the mean value, and we normalize using this mean value. It can be 0.35, it can be 0.60. So
Right. So that there's an assumption that the ChIP-seq data is, on average, flat on a big enough scale.
input,
Yeah, on the input. So I guess there's a certain scale that you can,
yeah. Mhm.
that there's a smallest sort of copy number variation that you can detect.
Exactly. There's a window.
So,
Yeah.
so what sort of scale are you sensitive
It depends
to?
on your coverage. So we have input data sets where we have 10 million reads, and then your window should be about 50 KB. If you use recent chip-seq data sets, when you have 20 million reads and more, you can go to a smaller window. And for example, if you use Nebula, then Nebula will automatically evaluate the window to use. The more reads you have, the better will be the precision.
Okay, thanks.
Nice talk. So actually, I have a question about this copy number correction. Is that since it's real copy number, so that means the gene in the copy number variated region should have 3 copies. So it should have 3 times of the signal, or 3 times of the enrichment.
Yes.
But after you fix it, it seems like it's just same as other genes.
Exactly.
So
Yeah.
that means this kind of effect just disappear from your result.
Yeah, exactly. So if you have a repression mark on one copy of the gene which is present in one copy, you will have 100%,
Yeah.
and we output 100%. If you have 3 copies and on all 3 copies you have signal, it will be still 100%. So you will have the same type of output.
Yeah, but does that just violate the biological truth? Because it's actually just 3 copies.
But if look, if you have this mark on 3 copies out of 3 repression mark, you don't have expression, right? If you have it on 2 copies out of 3, you will have expression from 1 copy which is not silenced. So if you use a method which doesn't correct for copy numbers, you will say, I don't have enough mark. You will say, I have a lot of marks. I have twice more than in 1 copy region, but still I have gene expression. And it can appear strange that I have a repressive mark, but I still have gene expression. Yeah? So, but if you normalize for copy number, you will say, I have my mark in 66%, and I have some gene expression. For me, it's like more, intuitively, for me, it's more correct. But it's up to you. So since you have this copy number information, just correct for it back.
Yeah. Another question about that is that if other people use FreeC to fix copy number bias, they use like Sizer, CCAT, or Max to call peaks, will they see similar results as yours?
I didn't try. Actually, CCAT, as I showed, it doesn't have copy number bias. It's just we believe that we do a better job than CCAT, but they don't have copy number bias. Their statistics are very stable in this sense. Probably, if you find a way to integrate into Max or Sizer, it will work perfectly, but just people didn't do it, and it's not my job to do it. I try to sell my tool.
Yeah, thank you.